are related, those are questions that you will have on Thursday are related to things that we're talking about in class. I want to make sure that one question that is going to be there, you had some chance to work with that kind of question, so it looks something like this. Right, so on Thursday when you see something like this, you won't be freaked out, you'll know what to do. So I'll explain that to you when we get to that part. Okay, just to kind of help you to think about some of the metabolism issues that we're talking about in this class. Okay, so there's a couple of topics we want to finish up with today for the lecture. So we're going to finish up chapter 5 today, and now there's a few more. One more copy, I think. So need a copy. There's a few extra copies around. Go ahead and go through this. Also, we're going to talk about some other types of metabolism, so particularly fat and protein metabolism. This is actually a time in the lab that we're going to be doing today, so you'll see the relevance of that. And there's a few other pathways that I'm going to briefly mention. Okay. So we'll have a worksheet. During the lab, I'll give you back your other worksheet, and then the lab today will be on enzymes. So again, it's going to be tight. It's going to be closely related to what we've been talking about in this chapter. Okay. Are there any questions from last time before we start going into this some more? Okay. So let me just kind of orient us to where we left off. Okay. So. This again is our overview of carbohydrate metabolism. Okay, this is what we've been working through the last time. We're going to finish up today. We've talked about glycolysis. Okay, now remember the important part of this is to know what goes in and what comes out. Okay, so glucose goes in. We get out pyruvic acid. So again, glucose has how many carbons? 6, right? Pyruvic acid has how many carbons? 3. Okay. All right, so the reason why carbons are important, as we discussed last time, is that this gives you a sense of how much energy is potentially left in the molecules that are in this metabolic pathway. And since there's still 3 carbon atoms left in pyruvic acid, there's a potential to get more energy out of that. And so we saw some of that going through some of the other pathways. So going back to glycolysis, remember some of the products that are important for in terms of energy. One is ATP, of course, which is our energy currency. The other is NADH, which is also known as reducing power. Yes. Okay. Right. So if it's reduced, has it gained electrons or lost electrons? Gained, right? We're going to see those electrons that it's gained play an important role later on in this process, okay? All right, as we started going through, because it's the electrons from NADH that get passed on through this electron transport chain. Okay? That's what allows us to make energy, which is what we're going to learn how that happens today. Okay? And so we talked about the Krebs cycle. All right, carbon dioxide is one product. That's actually where the pyruvic acid ends up. Okay, so we saw that we lost one carbon dioxide to go to acetyl-CoA. Acetyl-CoA is what goes into the Krebs cycle. Okay, and then we get out. That gets broken down into carbon dioxide. We get a little bit of ATP. We get more reducing power. Right? And this is a cyclical pathway, right? So there's ready to take in another acetyl-CoA to go through this process again, and that's how we're able to break down this pyruvic acid down to the one carbon endpoint, which is carbon dioxide. Okay? Now, I mentioned the electrons in NADH, also that's ADH2. This is the reducing power. This just goes into the electron transport chain, and when we going through this, we found that the electrons were being passed on from carrier to carrier. Okay? And as it gets passed on from carrier to carrier, that's what's used to generate ATP. This is oxidative phosphorylation. So this is the second process. Okay, the first process for making ATP is called what? substrate level phosphorylation, right? Substrate level phosphorylation. This is oxidative phosphorylation. This is what's done by the electron transport chain. Electrons get passed on until it finally ends up with oxygen. That's the terminal or final electron acceptor. Okay? All right, that's where the electrons end up. And in this process, ATP is made. The enzyme that makes ATP is called ATP synthase. Okay, so just to remind you about all the players that we have in this process. Okay, we talked about the electron carriers. All right, and now let's go into this process a little more deeply. Okay, so going back to this, right, what we see here is a flow of electrons from carrier to carrier to carrier. This in some ways is an energy graph, because as we go down this stairwell, this staircase, the energy of the electrons is decreasing. The reason why it's decreasing is that it's getting passed on to these electron carriers, and it's the energy that gets transferred from the electrons to these carriers, which is used by the carriers to make ATP. Okay, so we're going to understand how this process actually occurs. All right, so I'm going to try to show this to you in a few different ways. Here's one way, which is to describe it to you. What happens as electrons get passed on from carrier to carrier, okay, is that these carriers take away some of the energy, and it uses that energy to do this process, which is to pump out hydrogen ions. Okay, so what it does is that it takes that energy and it uses it to move those hydrogen ions from one side of the membrane to the other. So this is a membrane. Now, actually, if this were in your cells, what kind of membrane would this be?
Mitochondria.
Yeah, mitochondria. Okay, so this is a mitochondrial membrane that's inside your cells. In the bacterial cells, it's in the membrane of the cell itself. Okay, that's where this process occurs, right? And what it does in either case is that it pumps out electrons from one side of the membrane to the other. Okay. And when it does this, right, when it does this process, you're going to have an excess of hydrogen ions on one side, okay, and less hydrogen ions on the other, right? Now, just to kind of give you a sort of intuitive feel about how you can transfer energy, sometimes what's used as a model is imagine that this electron was a hot potato, literally a hot potato, okay? And let's say I took my pair of tongs, got this hot potato, passed it to the first person in front, and then told you to pass the next person, next person, next person. What's going to happen to that potato as it gets passed around the room?
It's going to cool off.
It's going to cool down, right? And what's going to happen to your hands as it gets passed on to you, to your hands? Right? If you have a hot potato in your hands, what's going to happen in your hands?
You get hot.
It's going to get hot, right? Because some of the energy from that potato is actually being passed on to your hands, all right? And then as it gets passed on, right, the hands are absorbing that energy. It's taking away that energy from that hot potato. That's exactly what's happening here with the electrons as it's being passed on from carrier to carrier. Okay? Each carrier takes away a little bit of that energy, and in some cases it's used to drive this pump to move these hydrogen ions to one side. Now, again, to give you kind of an intuitive feel for this, okay, when you see positive charges on one side, because there's a lot of hydrogen ions on this side, so you see positive on that side, you have less on this side, so you have a deficit of positive charges or a negative charge. When you see positive one side and negative on the other, what does that remind you of? What around your house does this remind you of? Huh? Or if you drove your car here, what in your car does this remind you of?
Gas.
A battery, right? A battery, okay? And that's one way to look at this. It's exactly like a battery, all right? What we're doing by moving positive charges to one side, hydrogen ions to one side, and having negative charges on the other, is that we're creating a little battery, okay? And because you're creating a battery, there's actually energy that you can release from this, There's actually energy in that battery that's stored that you can release from this, okay? And so I'll give you a thought experiment. I don't advise you to actually do this, but I'll give you a thought experiment to show that there is energy in this battery. We use a car battery as a model, as an example. You can go to a car battery, put your fingers, put your one finger on the positive side, one finger on the negative side, and you can experience that release of energy. There's energy stored there. Okay, right? All right. So some of you may have seen that happen with just using jumper cables. Okay. All right. And so there's energy stored in that battery. Okay. And what the last layer of this ATP synthase does is that it releases that energy by allowing those hydrogen ions to flow back through across the membrane, all right? And so that's going to release that energy just like touching the positive and negative ends of a battery. That's what ATP synthase does. And so it uses that release of energy to make ATP. Okay, so that's how this process works energetically. Okay? All right, any questions about that? All right, so again, this is something you kind of have to digest. You have to kind of think about and look at to digest. That's one way to understand this process. So what happens is that you create a hydrogen gradient. It's also called a proton gradient because hydrogen ions like this are basically protons chemically. Right? And then the ATP synthase releases the energy stored in this gradient by allowing those hydrogen ions to flow back through, and that's what's used to make ATP. All right? That energy that's released is used to make ATP. Okay? All right, so let me give you another way of looking at this. This is what you would find in your textbook. And here you can see in a little more detail what's happening. Here is the NADH. Right? So now NADH comes from what processes? So that we talked about metabolism. Glycolysis and Krebs cycle, right? So that's where the NADH comes from. So that's why those processes went through the trouble of making this reducing power, because here's where the payoff is. Electrons are donated from NADH to this electron carrier. The electrons get passed on. Okay? As that happens, you can see hydrogen ions being moved across the membrane. So now you have positive charges on one side, these pluses are. You have a negative charge on the other side. So now you have your battery. Here, just to let you know, this is where FADH2. Okay, that comes from the Krebs cycle. Okay, right. And that's where it donates its electrons. Okay. Right. We get this gradient. There's ATP synthase. It allows the hydrogen ions to flow back across to release that energy, and that's where we get ATP. And again, the final, where the electrons end up at what, where do they end up? Yeah, oxygen right here, okay, and to make water as the end product. Right, so oxygen is the final electron carrier, okay? All right, so that's why you need oxygen, okay? All right, any questions about this? Okay, let me try this. So I'm going to try, let's try this right now. Where'd it go?
Oh,
Here we go.
thanks, girl.
Okay, so here's an animation to show you this process. And what you'll see is, here's NADH, okay? And what happens is that NADH, this might not have been the best one to show you, is going to pass on the electrons to here, okay, to that carrier, all right? And then now what's going to happen is that those electrons are going to be passed on from carrier to carrier. So as we go through this, you can see the electrons being passed on to this carrier, okay?
Bless you.
Thank you.
And what happens is that as that happens, in some cases, the hydrogen ions are going to get pumped out. So in this case over here, you can see, so let's see here, you can see how these hydrogen ions get passed out right, across that carrier, okay, as the electrons get passed on. Right? Now, remember that in this course, we're not going to focus on the individual components, so you're not going to know the order of all these different components in the electron transport chain. Right? But what I do want you to know is just overall what's happening in this process, right? The hydrogens are being pumped out to create this gradient. Okay? And then here's ATP synthase. And so ATP synthase takes those hydrogens, moves them back across the membrane, and that releases the energy to make ATP. Okay, so that's what we're seeing happening around here. So here's the ATP, gets that energy release, and then it becomes ATP. Okay? All right? Any questions about this? Now, for those you have, I think that you can find these animations on Mastering Microbiology. Okay, so again, I've given you different ways of trying to look at this, and so if these animations help you, right, I encourage you to look at this just to get a better idea of what happens. Okay. Any questions about this? Okay, so now, so that was a way to animate this process that we have here. Okay, and just to point out again, you can actually see where this is occurring. Here is the membrane of the bacteria where it happens. Okay, so it's happening across the cytoplasmic membrane. Here it is happening in the mitochondria. Okay. Now let's take a look at some of the energy accounting going on here. All right, so when you go through glycolysis, okay, we learn that you get 2 ATPs. Okay, that's what you gain, 2 ATPs. When you go through the Krebs cycle, you get another 2 ATPs. Now you have 4. When you add on the electron transport chain, that final step, right, that's where you get actually the bulk of your ATP, 34 ATPs. Okay, so in bacteria, so this is a bacterial version, right, you get 30 ATPs. The bulk of them comes from the electron transport chain. So that's why this process is so important for a cell, is that you can get a lot of energy from that process. And now to step back from this, right, if you're a bacteria and you live in an anaerobic environment, how are you going to do this process? You live in an anaerobic environment.
No oxygen.
No oxygen, right? Oxygen is, why is oxygen important for this?
Because it's the final.
It's the final electron carrier, right? So that's why you need oxygen, okay? All right? Now we're going to find out that bacteria do have some tricks up their sleeve to get around that problem, okay? But in aerobic respiration, right, that's why we call it aerobic, because it needs oxygen. This is where the oxygen is needed, okay? All right? Now, let me add on to this. So, we talked about aerobic respiration. That's where the terminal electron acceptor is oxygen. Okay? And oxygen gets reduced to water, right? Because you add on those electrons. There are some bacteria, E. coli as an example, where they can do another form of respiration called anaerobic respiration. Right? So this is where the terminal electron acceptor is not oxygen. Okay, so now let me ask you this. Can we do anaerobic respiration?
Yes.
No, we actually can't. All right, because our respiration is aerobic. Okay, so we require oxygen. Okay, we can do anaerobic metabolism, so we can make energy anaerobically, but it's not through respiration. And we'll come to that, right, at that point. So we'll try to clarify that. Okay, but some bacteria can do anaerobic respiration where the terminal electron acceptor is not oxygen. Okay, so remember, power of respiration is all that energy that you get. So some bacteria have evolved the ability to do it without oxygen. Okay, so in fact, we think that maybe some of the early forms of bacteria on Earth and maybe on other planets like Mars, have developed respiration, right, without oxygen because that was the way that their environment was at the time, okay? And so this may actually be a leftover from that early part of Earth system, right? So E. coli, for example, can use nitrate instead of oxygen. So those electrons end up on nitrate and they become nitrite, okay? All right? Any questions about that? Okay, so that's another trick that bacteria can do that's special for this. Okay, so going back to this roadmap, we've talked about respiration. We've talked about three components of respiration: glycolysis, Krebs cycle, electron transport chain. Okay, we've seen how all the different products get used in this process. Okay, so we talked about electron transport chain. We looked at NADH and FADH2. Okay, and we learned about what the final electron carriers are, what the final electron acceptors are. And so that's why in this diagram they kind of kept it ambiguous, because bacteria can be more than just oxygen. Okay? Now, as you may notice, there is an entire part of this map that we haven't covered yet, and that's what we're going to do next, which is fermentation. Okay? So that's actually important. So I should probably write that down here, right? Because a lot of the lab experiments that you're going to be doing involve or are related to fermentation. Right? So this is actually an important process to try to get some exposure to now before we actually do it in lab. What happens in fermentation is that we go through glycolysis, right? So we're still making some ATP and NADH, but we have a different endpoint. You see right here, we actually make fermentation products instead of going through these other two processes. Okay? So in fermentation, you get low amounts of ATP because all your ATP comes from where? From what process?
Electron transport.
And that electron transport chain comes from. So if we go back here, where does all the energy come from in fermentation? Glycolysis, exactly, right? Glycolysis. Okay? All right? So that's what, and you only get 2 ATPs from that. So that's all you get, which is enough for a bacteria in many cases. Okay. Now, also in fermentation, we'll come back to this. You also make NADH, but the NADH doesn't get used. It gets regenerated to form NAD+. Okay? NAD+ is what feeds glycolysis to make NADH. Right? So you have this kind of cycle that goes on, where it doesn't get used, where the reducing power doesn't get used to make energy. It just gets turned back to NAD+. We'll see that a little more. Okay, so also this process doesn't require oxygen. Okay, so if you look here in this roadmap, oxygen isn't needed until here. This fermentation process completely ignores that, so you don't need oxygen. Also doesn't use the Krebs cycle or electron transport chain. Right? You only need glycolysis. Now, I point this out because in lab you're going to see this. There are 2 important types of fermentation tests that you're going to do. One is for mixed acids. So that's one example, one family of fermentation reactions that can happen, where the end product is going to be an acid, like lactic acid or acetic acid. So just to give you a context for this, lactic acid, fermentation that makes lactic acid, there's actually some food products where you have this. What would be an example? What would be an example of a food product where you have fermentation occurring and your end product is lactic acid? Where would you find, so lactic acid sounds like what kind of product? Milk, right? So what kind of milk product gives you? Cheese is an example. What else? Yogurt, right? Yogurt's another example, okay? All right, so in fact, if you look at, how many of you consume yogurt or eat yogurt? Yes, a lot of us, some of us. All right, so if you look at a yogurt container, you'll notice it says live active cultures, right? What kind of cultures are those? What kind of organisms are those? Yeah, they're actually bacteria, okay? It's bacteria that will actually produce lactic acid as a fermentation product. So they're actually fermenting your yogurt, okay? So as you let the yogurt sit and sit and sit in your refrigerator for longer and longer periods of time, what happens to the flavor of the yogurt?
No sour.
It gets more sour, right? That sourness is coming from the fermentation to make lactic acid, okay? Because those bacteria just are turning that that milk sugar, which is what? What's the chemical name for the milk sugar? Lactose, right? It's turning lactose into lactic acid. Okay? So I'm running through this with you right now because you're actually gonna be doing tests in lab where you're gonna be working with lactose, right? So lactic acid is one of the obvious products for that. There's another type of fermentation which is less commonly known, but still important, where you get 2,3-butadiol, okay, as your product. What you're going to learn about in lab is one of the intermediates that's formed in this process called acetone. This is, as opposed to acid fermentation, this is a form called neutral fermentation, where we don't make acidic products, okay? We make neutral products, and so this is a whole other class of fermentation reactions that you can actually measure in lab. You're going to do that. All right, so just to give you some examples, all right, here is acid fermentation, where we get different kinds of acids being formed. Here is neutral fermentation, or 2,3-butadiol fermentation, where again we go through glycolysis. Here are the fermentation reactions where we get acetone and then 2,3-butadiol. Okay? There's a test that you're going to do in lab where you're going to look specifically for this product, or you'll be looking for these products. Okay? And that's how you can differentiate between the two types of fermentation reactions, two major types of fermentation reactions that occur in bacteria. All right, I'll give you one more handle on this. This again is from your book, from a figure from your book. This shows you the different kinds of fermentation products that you can get using a more familiar spin to this. Okay, so you can see, for example, what kind of fermentation makes cheese, right? What kind of fermentation makes alcoholic beverages, right? You can even see one that makes acetone for nail polish remover, right? Or isopropanol for rubbing alcohol. Here's one for acetic acid. Okay, so all these are different examples of fermentation that occurs. Any questions about this? Okay, so now, before we